Hello everyone, welcome to our first lecture video on Unit 3, Cell Structure and Function. In this lecture video, we're going to focus on cell history with a few scientists, as well as just a quick description on the differences between prokaryotic and eukaryotic cells. So let's get started right away. All right, so if I click here and get our nice little movie here rolling, we're going to start with the first scientist, whose name was Robert Hooke. All right, so I say 1665. You do not need to remember these dates. They're just there kind of for a reference so you can kind of see where in history this was taking place. But here's a really creepy picture of Robert Hooke. Again, I had mentioned in class that this is a painting of him that was done years after his death, where everyone kind of forgot exactly what he looked like. And the reason for that was we had said that Isaac Newton had actually gone after Hooke's death and burned all of Robert Hooke's portraits because he had a massive feud with him over their careers. All right, so what do I want you to know about Robert Hooke? Well, he was the first person, at least he is given credit as the first person of producing and using what we call compound microscopes, all right, to look at cells. Now, what a compound microscope is, it's a microscope that is going to make use of two lenses at the same time, both what we call an objective lens as well as an eyepiece. So if you draw your attention over here to a picture of one of the microscopes that Robert Hooke actually made himself, you're going to have an eyepiece at the top, which you look at, and that's usually a magnifying lens that's no more than about 10x, which means it'll magnify something 10 times its original size. And then towards the bottom, you'll have another lens called the objective lens, which is what immediately starts to magnify the specimen that you put on the specimen holder or stage if you're looking at one of our modern day microscopes. So the image gets magnified by the objective, transferred up here to the eyepiece where it gets magnified again by whatever the magnification value of the eyepiece is. So let's say the eyepiece is 10x. And the largest objective on one of our microscopes in the classroom is 40x. So this specimen you're looking at would be 40x times 10x, and you would be seeing it 400 times its normal size, whatever you're zoomed in on. So you'll always multiply the eyepiece times the magnification of the objective lens that you're using to get total magnification. So when Robert Hooke made his first compound microscope, all right, and the first thing he looked at was cork cells. All right, so he took a thin slice of cork and looked at it under the microscope. And to him, and this is what it looks like up here, to him it looked like these tiny little rectangular rooms that monks in a monastery would live in. And those little rooms are called cells. All right, so cells aren't named after a jail cell or anything like that. They're actually coined from this term cell that describes the little tiny room in which monks in a monastery live in. And when you look at cork cell like this, really the only thing you're seeing is the cell wall. The cells are dead. Cork is the bark of a cork tree, right? And you're just looking at the cell wall, which is made up of cellulose that is very tough and can't really be broken down. So he saw these empty rooms, and he coined the phrase cells. Now, around the same time, so 1674, so the same time frame both these guys are living, we have Anton von Leeuwenhoek. All right, you don't have to spell out this name, you just need to be able to recognize it in multiple choice. Anton von Leeuwenhoek, he was big on making what we call simple microscopes, which are really just one lenses. All right, he would make kind of looks like a paddle down here, right? Something you would like grab and hit somebody with. But up top here is where you would put a specimen right here. And right where my cursor is, right, got my little laser pointer going, is the magnifying lens that he would have, again, made, just like Robert Hooke did. He would have made these lenses by hand. So you put the specimen here, and then you look through the other side, through the magnifying lens, through the specimen, and you have a light above the specimen, and you could get a really detailed image of whatever the organism was. All right, these microscopes that Leeuwenhoek were making actually had better detail than Hooke's. Hooke's could get closer up, but his lenses were not made as well as Leeuwenhoek's were. So Leeuwenhoek, even though he wasn't getting as close up to a specimen, he was actually seeing them a lot more clearly. Now, Leeuwenhoek liked to look at lots of weird stuff. He loved pond water. He would look at vomit, feces, urine, you name it, he would look at. But when he looked at pond water, all right, one of the first things he ever looked at, he was able to see actual cells like you see down below here, and he was seeing them move. Now, he might not have had as much detail as in these two pictures down here. This is definitely a mixture of pond water, and this would be an amoeba over here that you could also find in pond water. An amoeba is the largest single-cell eukaryote that you get, and we are eukaryotic cells also. He thought they were little tiny animals that he just couldn't see clearly because his lenses weren't good enough. So he coined them animalcules. So what he was really looking at were either large clusters of bacteria that were moving, or more than likely he was looking at very large single-celled eukaryotic cells. This amoeba here, you could probably see in a beaker with your naked eye, but it would just look like a little tiny like air bubble that wouldn't just be going up. It'd be going left, right, down, circles, sideways, you name it. You could actually, even with your naked eye, see it. All right, so that's what he was looking at. So he was the first person to actually see living, moving microorganisms, and he called them animalcules. Now, we're going to fast forward from 1674 to the 1800s, like almost 200 years, right? All right, 1674 to 1774, we're not quite to 1874 yet, but a long time where there's not a lot of advancement in what we know about cells. Part of the reason why is microscopes are very hard to come by, they're extremely expensive, and they're still limited in how well you can make the lenses to get a good magnifying power. You can't get to what we can today in the classroom of 400 or even up to 1,000 or 2,000. All right, so they were limited. Now, before I move on, let's just do a basic definition of what a cell is. A cell is what we call the basic unit of life. Think back to one of the first week of school when we went over the eight characteristics of life. All living things have to be made of cells, right? Even if you're just a single cell or you're a multicellular organism like you and I. All right, a cell is the basic unit of life. Now, we have coined the phrase cell. We can see that they're alive and moving under a microscope, but we don't really start to find out more about them until now, 1838, where two scientists, Schleiden and Schwann, all right, one a botanist, one who studies animals, a mammologist, studying all types of animal tissues and samples from numerous animals around the world, were sharing information that they had been accumulating through experiments over decades. And they established the first two components of what we refer to today as the cell theory. All right, remember what a theory is, though. It's not 100% truth. It's just the best explanation based off repeatable experiments that give us the same data over and over and over again. So these things could change in the future if we find alien life. Now, between the two of them, they came up with these two parts of the cell theory. And they sound kind of like obvious to us, right? That all living things are made of cells. Well, duh. And cells are the basic units of structure and function. So this isn't the definition. The definition is the basic unit of life. What this says is that pretty much they give you the overall structure and help dictate functions of the overall body. Now, all living things are made of cells is obvious because between the two of them, any of the thousands of plant species or thousands of animal species they looked at under the microscope, they were all made of cells and nothing different. So they had the evidence to back it up, so they stated it. They're the ones that give given credit. As well as cells are the basic units of structure and function. Think of it as, for structure, your whole body is supported by a skeletal system, correct? Well, guess what? Cells are responsible for producing the calcium that hardens to produce your bones, to give your structure. And then they also are used to create tissues, which turn into organs that serve a very important function in your body. Like the heart is very important, the liver is very important, you name it. All right, so they have very important components, or functions, I should say, within either a human body or plant structure. And then a couple decades later, we get a gentleman by the name of Rudolf Virchow in 1855. Again, these dates are when they would come out and publish their information. And for decades, he had been looking at plants, animals, you name it. And he was looking at both living and dead cells. And anytime he viewed these cells under a microscope, new cells only ever came about from current living cells. He never saw dead cells give rise to brand new living cells. So he came out and said, based on his data for decades, that all cells come from pre-existing cells. All right, that contradicted something called spontaneous generation. Spontaneous generation refers to the idea that cells can just pop out of nowhere over time. A lot of people believe this for thousands of years, actually, and some people even today. All right, they believe that cells can just pop out of nowhere. All right, what Rudolph was trying to say was, he wasn't trying to explain how cells were first created. All right, however you want to believe that, godly intervention, alien intervention, meteorites, asteroids bringing cells here to Earth. What he was saying that once cells first were created, however they were, from that point forward, they only came from pre-existing cells, period. All right, a lot of people were upset with him. He got death threats because it kind of went against religious belief, because people wanted to believe that, the God that they believed in was the one that decided whether or not cells were created. But he was showing that through his data that cells come only from pre-existing living cells. Now, those are the first original components of the cell theory. Today, we've added several more based on, obviously, the technological advancement we've had in the last 200 years. So, one of the new ones is we now know that all cells contain hereditary information. In other words, they have DNA. We also know, based on advanced technologies, that all cells, bacteria, plant, animal, fungus, you name it, they're pretty much chemically the same, meaning that they're made of proteins, carbs, lipids, nucleic acids. There isn't anything different in any of these cells. And finally, all chemical reactions that produces energy, as well as generic reactions that establish your metabolism overall in your body to maintain homeostasis, occur within cells. So what do I mean by that? So if you're going to break down glucose to make ATP, that happens in a cell. If you're a plant cell using sunlight to create glucose during photosynthesis, that occurs within a cell. All right. If you're the liver and you're made of lots of liver cells, and you're going to turn numerous glucoses into glycogen, guess what? That's happening within a cell. So these three additional, what we call the modern tenets of the cell theory, have been added based on our advancement in science over the last couple hundred years. So once more, there are numerous, dozens, hundreds of other scientists who've contributed to what we currently know about cells today. All right. However, these are the ones that we talked about today. These five are pretty much the main ones discussed in any college textbooks, high school textbooks, you name it, or any animation that you would look at. For example, I'm going to have you do an Ed puzzle on the wacky history of cells, and you're going to see numerous individuals, besides the one we just talked about, that have helped in some fashion to our understanding of cells today. All right, thank you. If you have any questions, make sure you ask me during our next class.